Good morning, everyone. Today I will be presenting my research project titled Smartphone-based Nanogenosensor for on-site detection of bovine tuberculosis using saliva as a diagnostic fluid. This research was conducted at Universidad San Sebastián in Chile. Bovine tuberculosis is a zoonotic infectious disease caused by Mycobacterium bovis, which significantly impacts productivity and competitiveness in the dairy cattle sector. In Chile, the intradermal tuberculin test is the standard method for a rapid screen of M. bovis, which is based on a delayed hypersensitivity reaction. Despite its low cost and ease of application, this test has several drawbacks that hinder effective control and eradication of the disease. These limitations include challenges in differentiating non-tuberculous mycobacteria and a delay in obtaining results, which are available 3 days post-inoculation, with diagnostic confirmation potentially taking weeks. Additionally, there is a variability in the immune response among cows, leading to false positive and negative results. The aim of this study is to develop a rapid on-site detection method for Mycobacterium bovis to enhance disease management with a genosensor based on fluorescent cadmium tellurium nanocrystal functionalized with a bioinformatically designed nucleotic probe. As you can see in the methodology diagram, the photonic nanobiosensor is functionalized with a probe specific for detecting Mycobacterium bovis DNA and includes a quencher that minimizes fluorescent emission. When it encounters M. bovis DNA obtained from saliva, the quencher is displaced and replaced by Mycobacterium bovis DNA. This displacement allows the fluorescent quantum dots to recover their fluorescence in proportion to the amount of DNA present in the sample. This fluorescence is then recorded with a smartphone camera and analyzed using an app developed by our lab called Quantum Dots Reader, which determines whether a sample is positive or negative to M. bovis. This table shows the result of M. bovis obtained from lymph node samples tested with tuberculin, qPCR, and genosensor. As you can see, there is 100% concordance among the methods, demonstrating that the genosensor is capable of accurately identifying M. bovis DNA. This graph illustrates the ability of the genosensor to discriminate between different non-tuberculous mycobacteria in artificially contaminated saliva. Our results indicated a specificity of 100% toward M. bovis DNA with a detection limit of 0.1 femtomoles, equivalent to 125 nanograms of M. bovis DNA, demonstrating sensitivity comparable to that of PCR assays. Finally, here is a timeline of diagnostic work developed using samples from a cow at our university. This cow was healthy at least until September 22. In September, she was released for free grazing, and 3 months later she was tested for the presence of M. bovis. The genosensor could detect this bacterium in early stages of the disease, as you can see in the rest of the timeline. In conclusion, the genosensor demonstrated the ability to achieve results comparable to those obtained when analyzing lymph node samples. Additionally, it was able to discriminate between different species of Mycobacteria in saliva. This case study illustrates that the developed nanogenosensor enabled the early identification of infection in the cow, which was later confirmed by tuberculin testing and qPCR. These findings represent a significant step toward developing a more efficient and faster on-site diagnostic method for bovine tuberculosis. This research was found by Fundación COPEQC in the project 2020 R79. This was my presentation. Thank you very much.